Hey, kiddos. Well, so a few minutes ago, we were talking about rhodopsin, right? And we were talking about the photopigments. Oh, you see, I changed, didn't I? My phone died, so it's actually the next day. But anyway, in the last video, we were talking about rhodopsin, and we had said that that rhodopsin is what we called a photopigment. And what is a characteristic of a photopigment? It's a pigment, it's a chemical that when it is activated by light energy, it changes shape. And we said we have two different shapes that it can take. It can be bent, that's what we call the cis form, or it could be straight, and that's what we call the trans form. And what did we say happens when it is in the cis form? When it's in the cis form, why did my computer die? Come on. Okay, it's back. When it's in the cis form, what it does is it causes the rod cell. We're talking about rhodopsin. All these studies were done with rod cells. We had said when it is not activated by light, it is going to be secreting a neurotransmitter substance called glutamate. And in this case, the glutamate is going to be inhibitory on the bipolar neuron. Remember, we had said we have the photoreceptor, the bipolar neuron, the ganglion, and then it goes to the optic nerve. And so coming forwards, when there is no light, the cis form of the photopigment causes the photoreceptor to secrete a neurotransmitter, glutamate, which inhibits the bipolar neuron from sending a signal. Then, when it's in the presence of light, boink, right? It goes boink, just like that. It goes boink, and it shifts from cis to trans. And when you do that, boink, when you do that, what happens is the inhibition is removed from the bipolar neuron, and it sends a signal to your occipital lobe saying, we have light. We can see again. There's light out there. And if you think about, well, how do you see? We had said it's like a TV screen. So you have little dots, and when you have light hitting this spot and not that spot, you get, oh, that's a puppy. You can see a shape, and that's how black and white vision works. Now, you'd said, well, you already covered that, and you are absolutely right. You're on the ball. But I mentioned that because now we're going to talk about color vision. And in color vision, this is going to be slide 23. So in slide 23, we had said that we have three different kinds of cone cells. Now, where in the eye do you have cone cells? The central fovea. That's right, the fovea. It's a location on your retina, which is where we have critical vision. And this critical vision, this is where the image is going to be projected, focused by the lens on the fovea centrale, and we have cone cells that are going to respond to different wavelengths of light. What is a wavelength? You can think of it as a color, right? So it's the a wave of light has this wavy form, and the wavelength is from peak to peak. It's literally a measure of distance. So this vibration has a distance in nanometers, and depending on how big that distance is, we perceive it as different colors of light. So when we look at the cones, we have three different kinds of cones, and they have photopigments, like that rhodopsin that I was mentioning. Photopigments that have different opsin parts. That's the protein part. And the different opsins will only respond to a certain wavelength. And you have one set of cone cells that only responds to the yellow-orange, and another that only responds to the greens, and another that only responds to the blues. And then, if you think of your television, you got a yellow spot with a green spot with another yellow, 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 blue, and you put it all together and you say, oh, it's a beautiful blue rose, you know, whatever it is. And it's like artist. You say you put this color with that color and you get, you know, violet. So if you have yellow and I'm trying to think, okay, yellow and something else, you mix the colors together is what I'm trying to say. So, all right, so if we look on slide number 24, you can see that we have this color spectrum along the bottom. I'll show you. There it is. So you can see that we have, and it says 400, 500, 600, 700. Those are distances in nanometers. So the blue light is found, blue light has a wavelength of 420 nanometers. That's what is being picked up by the S-type cone receptors. And then 531 nanometers, this is the M-type cone receptors, the greens. And then 558 nanometers, these are the L-type cone cells, and these would give you the reds. Okay, so what about color blindness, right? You've heard of people being colorblind. Well, what it is it means they have a deficiency in one or more of these cone cells. And so they can't distinguish colors as a result of that. And it ends up, just an interesting aside, 1 in 10 males are colorblind. That's a lot. Women, it's a sex-linked characteristic, and so it's much more common in men than it is women. And I remember when I was a young biology student, we had one of these books, and you could flip through all of these pages, and it was a test to see if you have color blindness, and there were probably 20 different pages. And you'd look at it and you'd see dots, but you could see a 63 or, you know, something in it. And my lab partner, this is when I was in college, he couldn't see them. And he got really twisted. He said, oh, no, I'm not colorblind. I don't know what he thought. But anyway, he was. But anyway, it's really common in men. Okay, so let's see what we have next. All right, so in bright light, like I am right now, my rod cells, boink, every single one of them is in the transform, right? So what would I see with the rods? Just light. You couldn't see anything, right? Because you have to have dark and light to see and to make out a picture, right? So in bright light, the rod cells are really doing nothing for me to allow me to see. But luckily, I've got those cones. And the cones in bright light, they're just responding like crazy, and I can see my green sweater and the light blue of my shirt and the ruddy reds of my face, right? So all of that, this is when my cone cells are in their most magnificent form, right? But what about when it's dark? They don't function at all in the dark. You have to rely on those rods. So we had this duplicity theory that says that no, it's in red, no single receptor type can provide low light sensitivity and high resolution. You have to have both of them. That's what we're saying. It's on slide 25. Check it out. Okay, slide 26. I'm going to show it to you. We have a cool picture here. Okay, so you can see we have, so what did we say? We had said that tracks go from one side of your brain to the other. Remember, we have that optic chiasma, right? It's where they cross. Well, it ends up, and we had said, we had the optic chiasma where they cross. And what did we say? We had said that when a nerve crosses from one side to the other, we said it's decussation, right? Well, in this case, it's not purely decussation. And if it stayed on the same side, what would it be? It would be ipsilateral, right? So the term we use is called Hemi Decussation. What? Hemi, it means partial. Some of the signals from my right eye go to the left side of my occipital lobe, but another half of it stays on the same side. And then same with my left eye. Part of it decussates over to the left side or right side, whatever, to the other side, and the other, whatever, it stays on the left side. So this term, hemidecussation, means that half of the fibers cross to the opposite side of your brain. Now, we have what we call stereo vision. Our eyes are in the front of our heads. Think about a bird. I hear. They don't have stereoscopic vision. If a bird wants to look at something, they go like this, right? Now, why is that? That's because they got all kinds of things trying to eat them. So they need to have a larger field of view. Now, so that gives them a larger field of view because their eyes are on the sides of their head. But the downside to that is they don't have as good of depth perception. If you have stereoscopic vision, by adjusting your eyes left and right, you can figure out how far away something is. This is what gives you the ability to have really good depth perception. The condition where the eyes are on either side of your head would be what we called panoramic vision. All right, that's all I'm going to say about vision, and we're going to come right back, and I will be right back this time, and we will be talking about hearing. Okay, see you in a minute.